Can you guys see that now? Okay, cool. There we go. So I just wanted to ask if anybody had any questions from the homework. You guys were asked to complete questions number 1 to 4 on page 20. And it says for number 4, you can skip the diagram component of that table. So you still do number 4, but you just don't do the diagram part. Does anybody at home have any questions from the homework? Or Katav, you said you had questions?
That was the question on diagram.
The question was like what to do for the diagram?
Yeah, I didn't understand. Like, I didn't know if you wanted me to do it or didn't want me to do it.
Just the diagram part you don't need to do because you don't need to know the shapes. Like I'm never going to ask you to draw a protein or something like that. So don't worry about that. But the other parts, what the function is of each, you do need to know that, like what are proteins. Okay, cool. Just so you know, guys, for the homework stuff, so not all of that is part of an assignment. But at the end, possibly at the end of this unit, I got to figure out how this is going to work. I'm going to get you to submit. You guys are going to kind of pick and choose some things from the unit that you've done and put it together as a portfolio. And I'm going to use that as part of your assessment at the end of the unit. So you'll be like, I did these homework questions, I did these homework questions, I did this lab. And like, these are the 3 things I'm going to choose to put in my portfolio. And then I'm going to look those over and use that as part of your assessment, okay? We're not going to do like a big formal unit test at the end of the unit. Instead, we are going to do a quiz tomorrow. So tomorrow, by the way, is the last day of the unit. I know it went fast. It's going to be fairly short. I think maybe around 20 marks. Okay, it'll be all multiple choice. So that's going to be part of the assessment from the unit is that multiple choice quiz. I'm going to do it during block 3, which means you write it at home. How long are you going to take to write it? That's up to you. Okay, so I'm not going to put a time limit on it. You can take as long as you want to write it. It's going to be very difficult for me to regulate like how people are writing this quiz/test because at least half of the people are at home anyway. So I have to keep it consistent for everyone. So I'm just going to get everybody to write it at home. I'm going to put the due date for this test as the day before you come back to this class. Okay? It's a long time. So what that means is you can write it right away tomorrow if you want. If you feel prepared, you can write it right away. And I highly recommend that you do that, okay? Everything will be fresh in your mind. You can do a quick review, then you can write your quiz, okay? I highly recommend you write it tomorrow. However, if you feel like you're unprepared, you can take some extra days if you want to look over the stuff from your notes to get ready for it. That's totally fine too. But you are going to be starting your other course number 2, on Thursday. And I don't know what you've got in course number 2. Maybe there's a lot of work you have to do for course number 2, so you may not want to put it off into course number 2 when you start getting super busy with course 2. I will leave it up to you though, okay? Some people want to take a little extra time to look over their notes. When you're writing, if you normally take a little extra time to write, don't worry about it, okay? Just take as long as you need to write it. I don't really care. The quiz is going to be an auto marker, which means you write it, it's going to tell you your results right away, and it's going to go straight into my mark book, okay? So that's one part of the summative component of this unit is we're going to write that approximately 20, maybe 25 mark quiz. It'll cover all of the topics that we covered in class. I'm going to post a list today after school of all the topics. Today we're going to be talking about cell membranes, diffusion, and osmosis. Yesterday we talked about, boy, we've been going so quickly, I don't even remember. Right, cell theory, components of cell theory. We talked about organization of living things. We talked about the macromolecules that are important for organisms, then their function. Okay, so like all those topics that we've covered in class are going to be part of it. I'm also going to ask some questions from the lab activities. Okay, so I'm going to take pieces from the lab activities as well. General knowledge stuff from the lab activities. If you've completed them and you're familiar with the material, you probably should be okay for those. Okay, so that's going to make up part of the assessment. The other part of the assessment is that we're going to be working on a project tomorrow. I'm still sort of deciding on the details for this, so it's probably going to be you creating a presentation for me and recording a presentation for me, okay? It doesn't necessarily have to be your face. Some people are like, oh, I don't want to put a face on camera. That's fine. So you can just have it as a Google slideshow probably, and then just talk. okay? That's fine as long as you're recording audio or whatever. So we're going to be doing that as part of tomorrow. I'm going to try and give you enough time tomorrow. The idea is that you do the entire thing tomorrow. It's going to incorporate some of the stuff that we've already done. Plus, it's going to probably be a research project on how you're going to pick a environmental factor. Lead, mercury, excessive heat. Pick something like that, and you're going to look at what the effect is of that thing, that environmental factor, on cells. So what happens when a cell is exposed to lead? Okay, it's going to be like a research project on lead poisoning. What happens to the organelles in the cell? What is what was the effect on cells of that environmental factor? We'll talk about it more in detail tomorrow, but in general, that's what we're going to do. Yes?
Will it just be focusing on human cells?
It doesn't necessarily have to be human. No, it could. I mean, you could, if you wanted, you could say like the effect of sunlight on plant cells or something like that. That's environmental factor and its effect on a cell. So it doesn't have to be human. The human stuff tends to have more information because there's more research on just effects on human cells. So, but you don't have to. I'll leave it wide open. Yes.
Quick question though. For the thing for Thursday, switching classes, if we're in cohort B, would we be staying at home or would we be going?
If you're in cohort B of your second course, you would be at home for the first half. I don't know how many days it is. I could look it up, but the first however many days of course 2. Yeah, so you might not be at school starting Thursday, depending on what cohort. And somebody asked this this morning as well, like which cohort am I in for the second course? It's not necessarily the same. So you might be in cohort A for this course, or you guys are on cohort B, or at home you're in cohort A. But you might not be in that same cohort for your second course. You should have got an email from your teacher from course number 2 telling you which cohort you're in. If you haven't, please email them today and follow up and figure out which cohort you're in, okay? Because you do need to know. You got to figure out if you're supposed to be at school or not. So.
I'm in B, so I'm at home.
Okay, so then you'd be at home. Yeah. So anyway, make sure that you actually do that. Were there any other questions from the homework? Okay, the other thing I wanted to mention was in terms of like, what do you need to hand in? People are asking me, like which assignments like have due dates and need to be handed in. So if you're looking for that information, if you look on here and you click on assignments at the top, I'm the assignments tab right now. These are the assignments you have to hand in. They all have a rubric associated with them. They have to be completed. I need these in order to give you a mark at the end of this course, okay? I will be going through and marking each of these. So you'll notice student information survey, okay? I have 13 out of 24. That's not great, okay? That was supposed to be done the first day of class. I don't have it from a lot of people. You have to complete that. I need that information from you. So for those of you at home, if you haven't done it, or maybe you have done it and forgot to turn the assignment in, please turn it in, okay? Same goes for you guys. If you've done it already, just make sure that it gets turned in. The cell biology vocab list, that's due on Wednesday, okay? So it always has the due date written right on there. So that would be like the last thing that you do for this unit. You're gonna hand that vocab list in, and I'm gonna give you a mark for that as well. But we're gonna keep adding to it. The cell types lab assignment is what we're going to be completing this morning during learning block 1. Okay, hopefully by the end of the day today, that lab is complete. I'm gonna give you almost 2 hours to work on it now. You should be able to complete it. It's quite a long lab though. And then the diffusion lab, we were going to do towards the end of today's period, but we'll see where we're at. This one might get bumped to the beginning of tomorrow as well. So we'll see, depending on where we're at, okay? But just keep in mind that if it's in this list, I need it from you. You have to do these things. This isn't like don't worry about it. And you shouldn't push these, okay? Try not to push the deadlines on these. You're gonna get stuck in course 2, focused on course 2. You're not gonna go back and finish this stuff from course 1. Like it's gonna build so fast, guys. We're already technically like on the calendar, we're a month into this course. That's where we are right now. So like try and keep that in mind. Yes?
Are you marking the organelles of the cell thing?
The homework?
Is that homework? Like this
Which organelles of the cell? That assignment? So you could potentially submit that organelles of the cell piece as part of your portfolio. That's later. I'm going to explain how that process is going to work. I have to kind of figure out how to mark the portfolios. I'm talking to some other teachers on how like we're going to organize that. But the short answer to your question is yes, I could potentially mark that. You could select that as part of what you're going to submit as part of your portfolio. Not to mention, you need to know everything on there for the quiz tomorrow. So you do have to do it.
You have to memorize them?
It's not memorization. I guess a piece of it is memorization. Yeah. What does a lysosome do? It breaks down cell waste and removes it from the cell. Do you need to know that? Yes, you do.
Is the test going to be in both or?
I mean, essentially, yes. You're going to be writing it at home, right? All of you are. So I'm not going to pretend that people aren't going to look at their notes during that.
Okay.
So I have to weight it a little bit less, and I'm going to weight the project that you're going to do tomorrow a little bit more. Does that make sense? Okay, so let me go over what we're going to cover today. So my content tab. Today is cellular biology day 3. There's the live stream at the top. We're going to pop back into the live stream again later. Well, I guess you guys here don't care, but we're gonna pop out of this, and I'm gonna give you an opportunity to finish the lab activity from yesterday. So that's learning block 1. We talked about that cell types lab. Yes, we didn't actually start it though. So for learning block 1, if you go back to day 2, because it is part of day 2, there's at the bottom there cell types lab assignment. If you open it up, oh, somebody's already done. There is a, how do I actually look at the assignment itself? Can I look at it? Doesn't let me look at it. That's annoying. Assignments. I don't want to look at submissions. I want to actually see the assignment. No, it doesn't. Okay, that doesn't make sense to me. Huh. Sorry, I'm still learning this too. Okay, well let me just look at what I've got written here for block 3. Okay, so or for block 2 from yesterday. So what you're gonna do is make a copy of the Google Doc. So you see right there it says make a copy of this Google Doc, and this is like a little highlight. You make a copy of that Google Doc. Okay, it's then it's editable if you make a copy, right? You can fill it in. And it's like a fill-in form. There's boxes where you write your answers in. You just click on them and fill them in. You don't need to use Cami for it because it's a Google Doc. And you're going to do that while you're going through the lab activity on Explore Learning. Okay, so you open Explore Learning. I'll do it right now so you can see it. I'm going to log in. And then the first lab activity that you're working on here in block 1 is the cell types lab activity, this one right here. So you open it up. And then the Google Doc, make me make another copy, just takes you through step by step, okay? It asks you a question, it gives you a spot for your answer. It asks you a question, gives you a spot for your answer, okay? And it'll tell you do this with the gizmo, do this with the gizmo. What happens? You know, you're just following along. The purpose of this lab is for you to get an opportunity to take a look at some different types of cells. We've talked about the different cell components, the organelles. We've talked about plant and animal cells, so you did that in the very first lab. This is an opportunity to actually look at some of those cells. I mean, look at them virtually, okay? The diagrams that they give you, which are drawings, are pretty good. They do really look like what these things look like under the microscope. So it's a fairly good approximation of this lab that we used to do in the actual lab. And the idea here is that you are looking for what are the differences between different types of cells. Here's a plant cell. What organelles do you primarily see? Which ones do you have the most of in a leaf cell? Which organelles do you have the most of in a muscle cell in animals? Which organelles do you see the most in an amoeba, a single-celled organism, that kind of thing. Okay, and the numbers of those organelles and which organelles you see are related to their function. So for example, in a muscle cells need lots of energy. What do you see the most of in a muscle cell? Mitochondria, the thing that makes the energy. Okay, the function is, the structure of the cell is related to the function of the cell. If you need more of a specific function of an organelle, then you have more of that structure. That's how it works. Okay, so it gives you an opportunity to actually see that in action. So that's the purpose of this lab activity. The second part of it, oh, it's not gonna let me scroll through, That's annoying. The The second part of it is you're going to be doing like a life test. There's a chemical test that you can look for to look for chemical signatures that show that a cell is alive. That's the second component here. So you're going to be doing just a little bit of biochemistry, and it's totally explained within the lab how that works. Okay, so that's the second component. When you're done this Google Doc and it's all filled in, you attach it to your assignment and you submit it. So that's during Learning Block 1 today. And like I said, it's an analog of the lab that we used to actually do in the lab, so it's pretty good. I'm glad that this exists at least. And then at the beginning of Learning Block 2, which was 11:05, is that right? So after the break, I'll come back to the stream, and then we're gonna do a little bit of a note together. Let me show you where that is. So then we're gonna be going into the actual day 3 stuff here. So where it says Learning Block 2, there's the note. It says it's available here. So you click on that, make a copy, open it in Cami. And that's what we're going to use to begin Learning Block 2. We're going to be talking about the cell membrane, how the cell membrane works, the different proteins that you find in the cell membrane, stuff like that. Okay, we're going to talk more about the microstructure of cells. There's a little video for you to watch, which is a summary of all that information. And there is a second lab assignment, but we'll see how long the note takes. So we may or may not do that lab assignment today. The last thing during Learning Block 3, and I'll mention this again, is I've got a really cool video here. I really love this YouTube channel. It's called Chubby Emu. And it's a doctor who just gives case studies. So they talk about real cases, real medical cases, and how they diagnosed those cases in the person and how they potentially treated that person. So this particular case study is about a person who drank too much water. It's a really interesting situation. It's about a person who drank 3 gallons of water in a short sitting. And it's directly related to what we're going to be talking about in our note. So I'm just going to get you to watch the video and then think about some things while you watch this video. There's nothing that you need to write up for it, but it is a cool video and they're extremely well done. So anyway, we'll come back to that later. So I'm going to give you guys an opportunity to, yeah, I think so too, Theo. I'm going to give you guys an opportunity to work on that lab activity. Do you guys have any questions in the room or at home about what you're supposed to be working on? Yes.
It's this one, right?
I have to come over there. I can't see that. I'm sorry. Even with my glasses, my vision's not that good. No other questions? You guys are good? You know what you need to do? If you have any questions while you're doing the lab activity, I'm going to leave the chat open here for this YouTube live stream. I'll put up a blank screen, but feel free to drop questions into the chat if you've got questions while you're working on this. And you guys obviously just ask in the room. Okay? All right, cool. Hey guys. Welcome back to the stream. We're going to be ready to go here in 2 seconds. So what you need to get for right now, we're going to put the lab on pause. You will have an opportunity to work on it again probably towards the end of this learning block. But we're going to do a little note together on a new topic. Just get this going again here. So the note that you're looking for, oh, not this one. The note that you're looking for is this one right here, cell membrane structure. If you go to the day 3 course content and then you look for Learning Block 2, it says the note is available here. That's what you want to grab. So you want to grab this, make a copy of it, and import it into Cami. I'll give everybody a second to get this opened up in Cami. Gonna turn the lights down here too so people can see the screen better. Katab, you know I can hear that from here, right?
Again?
Yeah.
Yeah.
Okay. Oh, you don't see the note. Okay. If you're looking for the note, it is on the content tab. So on bright space, if you click on content at the top, and then you got to select Unit 1, Cellular Biology, and then Day 3. Day 3 is today. And then Day 3 stuff will appear on the right. And then you got to scroll down until you see Learning Block 2. And then under Learning Block 2, it says, we will begin Learning Block 2 with a note on cell membrane structure and transport. The note is available here. And so then that spot where it says here, you click on that, and then you can make a copy of that and then open it in Cami. Did that help, Ayanna? Okay, cool. Are you guys good? Does everybody have it open? We're good? Okay, all right. Do you have it open, Katab? Katab?
Yeah,
Do you have it open?
yeah, I do.
The note? Okay. Okay, so we started off small in this unit. We talked about the organelles. We talked about some of the molecules that make up the organelles, the key ones. Those are the macromolecules, sugar, protein, and carbohydrates. Sugar, protein, and fats, and nucleic acids, and water. So those are the things that are primarily composing all of these different cell components. Today we're going to focus in on one specific organelle because it's a really important cell organelle because it controls what gets in and what gets out of the cell. So we're going to talk about that in the process of controlling what gets in and out. If you're looking for more information on this topic in your textbook, it's chapter 1.2 in your textbook. So this is kind of like a summary of what's in chapter 1.2. It's all about the cell membrane. So from this, what your takeaway is you need to know the 5 key functions of the cell membrane. So it does 5 important things for the cell. You should know what those are. We're going to go through them together, obviously. There are 3 important structures in the cell membrane. So there's like 3 components that make up the cell membrane. So you need to know what those are. It's membrane, phospholipids, membrane proteins, and cholesterol. We'll go through them together. And you need to know what each of those 3 things does in the cell membrane. Like what's the purpose for each of them. Then we're going to talk a little bit about how stuff gets across the membrane. So like why does stuff go into a cell? Why does stuff go out of the cell? How do you move things in and how do you move things out? Okay, so we're going to talk a little bit about diffusion, which you may have covered in grade 10 biology. Do you guys ever talk about diffusion?
Yeah, a little bit.
A little bit. Okay, so we're going to talk about how diffusion works. Ideally, we were going to do a lab activity today on diffusion, but I think that's probably going to get bumped till the beginning of next, which would be tomorrow in the morning. And then we're all going to talk a little bit about osmosis as well. Osmosis is basically the same thing as diffusion, but it's water moving instead of something else moving. So we're going to talk about those 2 things. And then lastly, we're going to talk about specifically what does the cell do to get stuff in and get stuff out. So those are, that's like how the cell transports materials. So you guys should know the different methods. So I have these broken down into the note under headings. So the first part is the structures. That's the first little part right there. And then the second part is what are the functions? I actually reversed the first 2 there. Then we're going to talk about the functions. So it's broken down into these like learning objectives, just so you know, if you're going back to review. Okay, so we previously mentioned, we talked about what fats are used for in the cell. We talked about how the main use for fats in the cell is energy, right? You eat fat because you need energy. But the other thing that it's used for is to actually make the cell membrane. The membrane is mostly made of fats. That's the main component. And this is the structure of them right over here. So if you look at the molecule, it's a big long chain of carbons. with some hydrogen stuck to it. So it's like two big long carbon chains like that. When you have a big long straight chain of carbons, they don't have a charge. So they evenly distribute their electrons. This is kind of going back to grade 10 chemistry a little bit here. So you don't have a negative end or a positive end. Water really, really likes negative ends or positive ends. It sticks to things that have charges. So if you have a super long thing like this with no charge, it actually means that it repels water. So this end of this molecule is actually going to push water away from it. It doesn't want to go near water. So another word for that is you say that it is hydrophobic. So this end of the molecule down here is the nonpolar end, and it hates water. So hates water is the same thing as saying hydrophobic. Phobic, afraid of water. So this end of the molecule is pushed away from water. The other end has a phosphate group on it with a charge on it. Okay? So because it has that charged group on the other end, charges are attracted to water. So the other end of this molecule is the polar group. Polar means that it has a charge. Okay, so it is a charged end, and that means that that other end, the head, is attracted to water in this fat. So that's called hydrophilic. Hydrophilic means loves water, loves H2O. Hydro. Oh, I always spell hydrophilic wrong. I want to use a Y, but it's an I. Hydrophil, 1L. Hydrophilic.
Can you repeat what you said about the polar head thing that you said?
So there's two ends to this molecule, to this fat molecule that makes up the cell membrane. There's one end that doesn't have any charge to it, that's the nonpolar end, and it's hydrophobic, it pushes water away. And then the other end over here is hydrophilic, it has a charge, and so it sticks to water.
Yeah, I understand that.
Okay.
There's something else you said about it. The polar head one. Something you said, I forget.
The polar head one? So this is the head. So
it
that.
sounded important, I forget though.
Uh-oh. Okay, maybe I'll say it again here as we move forward. Okay, let me know. So this is sort of generally how this molecule is summarized. Obviously this isn't like drawing the full structure, but they often use like a little circle to show you the head. And then there's just like these like two tail legs at the bottom right here. One of these big molecules right here is called a phospholipid. It's a phospholipid because it has a phosphate on one end, that's the top, and then the bottom part is a fat, a lipid. So it's a phosphorus and a lipid, phospholipid. Yes?
Instead of writing that there, can I write that above it?
Sure you can, absolutely. So obviously a cell membrane is not made out of a single molecule. It's made out of a whole bunch of these that are arranged into a pattern. So here's an example of a cell membrane here, and you can see that it's a whole bunch of these heads with their little tails. I'll draw them in a different color so they're easier to see. With their tails sticking down. And then there's another group of them on the inside of the cell. And they've got their tails facing into the center. So the reason that they're like this is because the head, that's the red part on here, is attracted to water, right? And that's what's outside the cell. So there's like a bunch of H2O out here on the outside of the cell. So this head right here is attracted to H2O, and so that's why it wants to face out. The actual molecules themselves are just kind of loosely sitting in the cell membrane, but they always point in that direction because the part that loves water is going to face the water. It just naturally wants to face that way. And then the same thing is true on the inside of the cell. So on the inside of the cell is full of water. more or less. The cytoplasm is mostly water. So you got water on the inside and water on the outside. So you also have that attracted to water part facing into the cell. And then you get this part in the middle. This part, whoops, don't want to make that quite so big. You got this part in the middle where the tails face inward towards each other, because they're the nonpolar parts. They're getting pushed away from the water, and so they end up sticking into the center. So the layer, the cell membrane that goes around the outside is, it has two layers to it, right? It has like an outside and an inside layer of molecules. And so then this is usually called a lipid bilayer. So collectively, this together is called a lipid bilayer. So the reason it's called a bilayer is because bi means two, right? Like a bicycle or whatever, two wheels. Okay, so a bilayer just means it has two, it's a two-layer layer, okay? So that's the basic structure of the cell membrane, is it's made of fats with a little phosphate stuck to them, and then they're lined up in this layer around the outside. But it's like a very loosey-goosey layer. This isn't like strongly attached. So if you poke on it with a part of some kind, a stick, or a little molecule or whatever, it'll bend and conform to that. Like it's really loose. So it's not really like a balloon, it's more like a bubble. In that, like, you know, the sides of the bubbles are like very flexible. So it's just like that, it's just like a soap bubble, really. So that's the main part of it, is the lipid part, the fat part. So we mentioned above here that there's three key structures, right, that are in it. So the phospholipids, that is the fat part, makes up the bulk of it. That's the actual layer. It's composed of fats. But then you need all the other functional parts. So that's the fat part. It also has a whole bunch of, oh, can you see it in this diagram? I don't know if it's actually shown here. Oh, yes, it is. There you go. There's one right there. It also has a whole bunch of cholesterol sticking into it. So going back to our lipid bilayer on the previous page, this isn't a totally accurate lipid bilayer, because if you had, what's actually here is a cholesterol molecule here, a cholesterol here, a cholesterol here, cholesterol here, cholesterol here, and there's a bunch of cholesterol that is just interspersed, little cholesterol molecules that are all over the place. You guys have heard of cholesterol before? You like get some in your diet, right? And like some people have high cholesterol. If you have too much cholesterol in your blood, it's not good for your blood vessels over time. It causes damage to your blood vessels. But you do need it. You do require some cholesterol because it sits in the cell membrane. And the reason you have cholesterol in there is to make sure that the lipid bilayer stays soft. If you have no cholesterol in your lipid bilayer, those little phospholipids are stuck together too tightly. And if they run into other cells and stuff like that and get pushed around, it causes the cell membrane to break. It ruptures, it falls apart. So you don't want your cell membrane to be like literally like a soap bubble where it just gets popped, right? It gets like a little bit too much pressure and it just explodes. So the cholesterol helps keep it kind of loosey-goosey. It keeps it from getting too stiff. So I think I wrote that on the next page here. Yeah, that's right. It causes an appropriate level of flexibility. It lets the cell membrane flex and bend around stuff. Especially important in animal cells. If you're a single cell organism, you need to be like have a really flexible cell or you'll just pop. So that's what the cholesterol does. The last component is you have to have a way to get stuff into the cell and to get stuff out of the cell. So you have to have stuff in the membrane, basically tubes. You have to have some tubes in the membrane to let stuff in and to let stuff out. So that's the third component here, which is the membrane proteins. So membrane proteins are proteins. They're made of protein, and they allow stuff to come in and go out of the cell, or they pump stuff in or pump stuff out of the cell. So if going back to our diagram over here, so we've got our lipid bilayer, we've got our cholesterol. I should have labeled that. This is supposed to be, I should use yellow to label. Let's call this cholesterol. And we've also got, what's the color I haven't used? Green. We've also got proteins that go through the membrane and make like tubes, basically, that allow stuff to go through. So there might be a tube that lets water come in and out. There might be a tube that is specifically designed just to let in only one type of thing. Like maybe it's a specific shape that only lets in a certain type of ion. Like maybe it only lets potassium into the cell, or maybe it only pumps sodium out of the cell or something like that. Okay, so these proteins are very specific, and there's a whole bunch of different kinds of them for moving stuff in and out of the cell. So these are called membrane proteins. So those are the three components. You got your lipid bilayer, your cholesterol, and your membrane proteins for transport. What you end up with is this crazy hodgepodge of all kinds of things sticking in and out of the membrane. You got proteins coming through, you've got sometimes there's sugar sticking out. Those aren't particularly important for what we're talking about. You've got cholesterol, you've got this lipid bilayer, and all of the components are kind of just floating around. They're very loose. So for example, this lipid right here won't necessarily always be in that spot. It's kind of like free to move around on the surface. So it might like kind of migrate over to this spot. Or you might have like this membrane protein kind of like float over here, okay? They kind of just move around. It's very liquid. And so because it's so liquid and like can move around so much, this way of thinking of the cell membrane is called the fluid mosaic model. So mosaic because it's got all these different parts that make up the cell membrane, and fluid because it's fluid, like the parts can kind of shift and go all wherever they want, okay? So whenever you hear about the fluid mosaic model, that's just this idea that there's just a whole bunch of stuff stuck in the membrane and then it just floats around and kind of goes wherever, okay? So there's some other stuff here that's mentioned in this diagram, things like glycoproteins, those are sugars. Don't worry about this. So this is not from this course, and you don't need to know anything about those, okay? If you want to take grade 12 bio, we can come back and chat about that another time. But let's just focus on the three major components. So you got phospholipid bilayer, transmembrane proteins that are channels and things like that, and cholesterol. Okay, those are the three biggies. So together, these things work together to do the 5 primary functions of the cell membrane, okay? The first primary function is it keeps stuff out, okay? It's a wall. It keeps stuff from getting into the cell. What are some things that the cell wants to keep out? What would the cell not want to come inside? What do cells not want in there?
Carbon dioxide.
Bacteria. Carbon dioxide. These are great. Viruses. Is it virus says like this?
Yeah.
That looks wrong for some reason. I guess that's right. What about waste?
Oh, yeah.
So cells are pumping out their cell waste, uric acid and urea and things like that. Those are being moved out of your cell, so you don't want that waste to come back in. What else?
Don't they take out like sugar and stuff? It's just too much.
Releasing sugar. The liver does that. The liver releases sugar into the blood for the muscles and stuff to use. I wouldn't say that it's necessarily an unwanted material though that cells are trying to keep out.
If it does like too much.
Then you're right then it would be in that case. That's like a special case, so I'm not going to write that down here. That might be a little confusing. But you're right though, sometimes cells don't want to take in more sugar. And then so you release a hormone that tells cells not to take up sugar. Not insulin, but the other one. What's the other one called? Glucagon? Does that sound right? It's not glucose. Glucose is the sugar. I can't remember what that hormone's called now. The other one.
I don't know. I was gonna say something else.
What was your question?
I was gonna ask, does it exert unwanted, like, like if there's like too much energy, does it exert that or no?
If there's too much, what do you mean by energy? Like too much sugar or fat?
Yeah, or stuff like that. Or is that basically what we were just talking about?
Well, it can restrict the entry of sugar. If there's too much sugar, it can prevent sugar from coming in.
Alright.
Yeah, definitely it can. Yeah, so sometimes sugar is wanted depending on the material. Like muscles might want to take up sugar, but maybe like your brain cells already have enough sugar, they can restrict entry for sugar. Definitely. Okay, it also prevents things from getting out of the cell that the cell doesn't want to get out. So what are some things that the cell would want to keep inside? It doesn't want to let it out. I'll give you one, the one that I can think of right away. Organelles. It really doesn't want those to leave, so it's got to keep them in. What else does the cell want to keep inside, prevent them from leaving? Like what?
Energy.
Energy, sure. Energy molecules. So that would be that could be sugar, or the cell makes something called ATP, adenosine triphosphate. It's actually discussed in your lab. You want to keep that stuff inside the cell so you can use the energy. That's the cell's most important energy molecule.
What would protein be?
Absolutely. It wants to keep its proteins inside. So it has intercellular proteins, proteins that are inside the cell that it doesn't want to escape. It needs them inside the cell. What else?
Oxygen.
Oxygen. Just want to keep that O2 in that it takes in, not lose that. It gets used pretty quickly, but okay, that's a pretty good list. So those are some materials that the cell wants to keep in, and that's what the cell membrane does. It keeps that stuff from leaving. It also allows communication between cells. So some of those proteins that are going across the membrane allow the cell to talk to cells that are nearby. Like the cell next door says, oh, there's an injury over here. You need to make a copy of yourself to fill in the gap, or whatever. Cells can communicate to each other through these proteins. They can send a signal. So, communication between cells. You could tell the cell to reproduce. It's mitosis. Tell the cell to apoptose. Apoptose is the opposite of reproduce. It's telling the cell to commit suicide. So the reason that why would you want a cell to tell a cell, it's time for you to back it up?
Used too much, or
It's what, sorry?
is it used too much, or worn out?
You're on the right, it's worn out, exactly. So the cell is like too old, right? It's not functioning properly. So you'd want to tell a cell to die if it's not functioning properly. There's a new word for your list right there, apoptose or apoptosis. That's when you tell a cell it's time to die. So if cells are not functioning properly, you want to tell them to die. If one of your white blood cells notices that a cell is making virus particles, maybe you want to tell the cell, better pack it in. You're about to explode with virus particles. We need you to shut your cell down. So your immune system can do that too, if it notices a cell is making a bunch of virus particles. And so that's just two examples that you could communicate with another cell. You could tell cells to do all kinds of things. You could tell it to take up sugar. You could tell it to take up some other hormone, or be more sensitive to a signal from another cell, or whatever. There's all kinds of different cell communications that could go on, but there's just two examples. So the cell membrane allows for communication. You can talk through it. It allows you to transport stuff into the cell. So it doesn't just keep stuff in, but it also lets you bring stuff in. What would you want to bring in? Think proteins? Okay, so you're not going to bring in full proteins, but you're going to bring in the stuff that you make proteins out of. Do you remember what those are called? The base unit that proteins are made out of. Anybody remember? So they're called amino acids. So amino acids are what makes up proteins. And yes, you want to tell your cells, bring in amino acids, transport them in. We got to use them to make proteins. What else do cells need inside of them? You gotta get it in there, transport it in. Think about what you need to eat. Sure, so what's in your food that the cells need?
Nutrients.
Nutrients, okay, so I'm gonna say micronutrients. Micronutrients are things that you need just tiny quantities of, like iron, or calcium, or what's something else you need in like a small amount?
Sugar?
Oh, sure, that would be outside, that's a macronutrient. I'll close my bracket here. So those would be micronutrients. You also need to bring in your macronutrients.
So what's
Sugar?
the micronutrients there?
Micronutrients are needed for chemical reactions in your cells, but you only need like a tiny little amount of them, micrograms every day. So for example, like you need to take in some iron, you need to take in some calcium, you need to take in some selenium. These are things that you need in your diet, but like just a little bit.
And then the macro, just like the bigger amount.
That's right, you need a ton of those every day. So sugar would be a macro, it's something you need a lot of, carbohydrates. Amino acids would be the protein part, and then you also need to take in fats, right, lipids. Okay, so that's the nutritional stuff. There's also metabolic stuff that you have to get into your cells in order to do chemical reactions, especially for your respiration inside of your cells. What do cells need to breathe in order to like make energy and stuff? Mitochondria needs these. What do you breathe in? Oxygen. You gotta get oxygen into your cell. So you gotta get oxygen in. I'll add one more in here. Sometimes you need to bring water into your cells. That's not an exhaustive list, but that's a lot of the materials that you need to pump in regularly. And then lastly, there's stuff that you want to get rid of. The removal of products. What do you want to get rid of? Carbon dioxide. So that's a big one. Your cells are always producing that because of respiration. What else? You generally are using the fat inside of your cells to build things or for energy.
But not good fat, you know what I mean?
You mean if you have like excess fat?
Yeah.
If there's excess fat in your blood, your cells won't take it up. So they don't usually need to get rid of fat, they just won't take it up if there's too much, and so it'll get sent to a fat cell.
Oh.
Your fat cells will absorb it and they store it.
Oh.
So in general, your cells don't take up the fat if there's too much in your blood. They don't usually need to get rid of it. Go ahead, Kadam.
Waste.
What's that? Waste products. So that would be all of your cell waste products. Your cells make waste. They break down protein, they create urea.
Jamie Fowler
That's loud.
to the custodial lunchroom. Jamie Fowler to the custodial lunchroom, please.
So those would be some examples of things that your cells are always trying to eliminate, remove from your cells. Okay, so how do you get them in and out? So this is something that you may have talked about last year or talked about in a different course. This is probably one of the most important principles in biology, which is the idea of diffusion. And diffusion is really a very simple idea. All it means is that if you have a high concentration of something in a solution, like in a liquid, the particles will spread out. That's all that it is. So things don't stay together in a big bunch. If you went to the far side of this room and you sprayed some perfume in here, okay, just sprayed it into the room, you already know this, that the perfume isn't gonna sit in a cloud in one side of the room, right? It's just gonna slowly spread out into the entire room. It's gonna go from a high concentration in the corner to a low concentration and spread out through the entire room. That's diffusion. That's all there is to it. So things always move from high concentration to low concentration. And that direction of flow from high concentration to low concentration is called a diffusion gradient. And materials always move in the direction of the diffusion gradient. So for example, if you had a liquid, let's say some water, and you dropped in a sugar cube or something, okay, and the sugar cube is going to dissolve, the sugar is not going to stay together in a little clump of liquid sugar inside the water, right? So these molecules are going to start bumping around And eventually, just due to the natural force of diffusion, you're going to get a distribution of these particles evenly throughout the solution. Okay? Things want to be just distributed normally. So, if you have a cell, draw my cell right here, and you've got a whole bunch of a certain particle on the outside of the cell. Let's pick something that could be on the outside of a cell. Sugar. Okay, actually, no, the cell membrane's not permeable to sugar. Let's pick something that can just go across the membrane. What can go across the membrane easily? Something that can go across a cell membrane. Mercury. You don't want this to go across your cell membrane, but mercury in certain forms is fat soluble. Okay? So let's say you got a bunch of mercury outside your cell. Hopefully you don't. So if you have a lot on the outside and the cell membrane will allow mercury to pass across it, and it does in real life, what ends up happening is you just naturally get migration of these things across the cell membrane until you have a balance of those two materials. Whoops. Until you have a balance of those two materials across the membrane. Okay? They're just going to want to equalize naturally. And there's actually a pressure for them to move in this direction from high to low concentration. Okay? That's diffusion pressure. Along the gradient. So the gradient just means the direction that the pressure is flowing. So in this case, the direction, the gradient, would be from outside the cell, inside the cell. So this only works if the cell allows whatever the material is to pass across the membrane. Mercury is a good example because mercury can pass across the cell membrane. But there are some materials that the membrane doesn't let across it, like sugar. So if you have a high concentration of sugar on the outside and a low concentration of sugar on the inside, there's a gradient pushing the sugar in, but there's no holes. There's no way for the sugar to get in, and so that you don't get a change in the concentration of sugar. Not purely based on the gradient, okay? Because sugar can't just go through. So that's the idea of diffusion. Things are going to naturally spread out until they're balanced on both sides of the membrane, if they can get across the membrane. Does that make sense? We're okay with the idea of diffusion here? Okay, osmosis is the exact same thing, except now we're not talking about the solvent, or sorry, the solute, that is like the solid stuff in the liquid moving. Now we're talking about the flow of water. So osmosis is diffusion, but for water. So if you have a situation like this in a cell, where you've got a lot of solute on one side of a cell membrane and not very much solute on the other, but the solute cannot move across the membrane, okay? In this case, if the solute could move across the membrane, like the red parts here could go across the membrane, what would happen if the red things could move back and forth? What would happen to this solution because of diffusion? It would even out, right? Some of these red ones would just go through the membrane, right? And then it would balance so that you had the same number of red ones on each side. But the cell membrane doesn't let those things go through the membrane. But it does let water go through the membrane. So instead of the red things moving from one side to the other, you're going to get water moving to try and make them the same concentration on both sides. So what ends up happening with water is actually going to get pumped through the membrane until the concentration is equal on both sides. Okay? So this is actually the exact same thing as this, but instead of having the solute move, which is the red stuff, in this case the solute can't move, so the other part has to move in order to balance it. That's the water part. So that's osmosis. This is really, really important in biological contexts because, for example, let's say that this was the inside of your cell right here, okay? So this was the inside of the cell. And this was the outside over here. So you've got a really high concentration of something inside your cells, and you've got a low concentration outside the cell, and all of a sudden a whole bunch of water moves into your cell. What's going to happen if a whole bunch of water suddenly goes into your cell? What do you think? Imagine if it's a water balloon, like a cell, and all of a sudden you pump a whole bunch of water in. It's going to pop, okay? It causes lysis. It causes cells to explode, basically, okay? So you don't want this situation to happen inside of you. You don't want your cells to have a huge amount of solute inside them, dissolved solid, causing water to flow rapidly into your cells. If this happens in your brain cells, it's really dangerous because your brain will swell, your cells will get larger in your brain, and your head can't get any larger. This is maximum size. So if your brain gets larger inside of your head, you can squish your brain cells against your skull and it causes them to die. It's really serious, okay? And this can happen in other areas in your body too. It can happen in muscles and things like that, depending on what's happening chemically inside your cells. So you need to balance solute inside and outside of the cell to regulate water movement so that there isn't water going in and out of the cells inappropriately, okay? This is gonna come up in the video that I have for Learning Block 3. Remember that medical case study that I mentioned? So in the medical case study, they talk about a person who drinks a whole bunch of water, and when that person drinks a whole bunch of water, the exact opposite is going to happen. So now what that does is it means that there is going to be not something different. It's this exact thing, which is that you're creating a very, very low concentration outside the cell of your solutes, right? Your sodium, your potassium, really low concentration because you put a ton of water into your body. And now inside of your brain cells, you're going to have a relatively high concentration of those things. And then this exact thing will happen. Okay, so that's a hyperhidrosis. When you take in way, way too much water, you actually create this situation inside of your brain cells. And then it talks about what the physical consequences of that are in the video. It's really cool. Yes?
How much is too much water?
Your body can process about 2 liters an hour, assuming that you don't have any excess water loss. So that would be that you're not sweating. So that's just through your kidneys. You can process about 2 liters of water an hour. If you're sweating, a regular like Nalgene is about 1 liter. So you can process about 2 of those an hour without sweating or increasing your output. Now, if you get a little bit more than that, like a tiny increase in the volume of water in your blood is not hugely problematic. Like you're not going to die from that. But if you take in 5 gallons of water or something like that, it's way more than your kidneys can process. And then you start getting into trouble when you do stuff like that. So it would be way, way more water than you would ever want to drink. You would have to force yourself to drink it. If you're thirsty, you're good. You can never harm yourself by drinking when you're thirsty. Yes?
Wait, so that's basically, I know like it doesn't really matter for like drowning. Like drowning, you're choking on the water. But
It's going in your lungs,
also,
yeah.
It's going into your lungs and also doing exactly that, right?
That's a slightly different situation when the water is going into your lungs. And it's also preventing you from being able to breathe
Yeah, that's
when it's in your lungs. But you're right in terms of if you have water sitting in your lungs, it does cause a problem with diffusion in those lung cells that can inhibit your immune system and cause infection and things like that when you have water sitting in your lungs. But we're only talking for small amounts of water. Once you get a lot of water in your lungs, you won't be able to get enough oxygen into your blood. You'll suffocate.
Yeah,
So that's what happens when you drown.
yeah.
So that's the far more problematic part, is that you can't breathe. Anyway, watch the medical case study video in Learning Block 3. It's really cool. So it talks about like the actual effects of this on a person, like what that would look like in a human. Because people do this sometimes, right? You know, this was in the news like a few years ago in Australia. They had that water drinking contest or whatever. It was like a radio contest. Did anybody hear about this? And like a person died because they drank too much water. And anyway, I'll let you watch the case study. It's really cool.
Also, drinking too much water can dilute your red blood cells, right?
Diluting the red blood cells
It can,
is not the main problem.
Or it does something to it, right?
You are diluting stuff.
You're diluting it because you're drinking way too much water, and you're getting more water in your bloodstream.
Right. So the thing that you're diluting, these red solutes in your blood are things like, the main ones that you're concerned about are sodium and potassium. So I'll write them on here. You don't want to dilute those ones in your blood too much. You need a certain concentration of sodium and potassium for your nerves to work properly, to prevent this from happening to your cells and causing them to explode, et cetera, et cetera. So you don't want too much water in your system. But again, under normal circumstances, you would never have this much water in your system. Oh, that comes up when people take ecstasy sometimes. So sometimes when people take ecstasy, it gives you like this unquenchable thirst. So people like drink a whole bunch of excess water, and then they also end up getting hyperhidrosis. So this is sometimes a side effect of taking ecstasy, which is interesting. So this is the same thing. I should also mention, by the way, that if you're sweating a lot, this is not as important because you're removing a ton of more water by sweating, right? So the actual amount of water that you need might be much, much higher than 2 liters an hour. So just so you know. Okay, so anyway, those are two concepts here, the idea of diffusion, solutes moving to spread out, and then you've got osmosis, which is the water that's doing the moving, but it's also spreading stuff out. Okay, it's the same thing. Okay, this all brings us back to what cells need, okay? What they want to do is they want to keep their internal conditions constant. Inside the cell wants everything inside to always be the same. In order for its chemical reactions and everything that it needs to survive to work properly. Same temperature, same amount of sodium, same amount of oxygen, same amount of carbon dioxide, always the same. Okay, that's the idea. Keeping everything the same inside of your cells is called homeostasis. Did you guys talk about that in grade 10 biology? Okay, so this is technically supposed to be in grade 10 as well. Not everybody gets to this part in grade 10. But homeostasis is always keeping the conditions the same. That's what your body is shooting for. You're trying to keep your body temperature the same. You're trying to keep the amount of water in your body the same. All of your systems are working together to keep the conditions the same, because that's the ideal condition for your cells to live. Your cells, most of them, are sitting in liquid. Not the extreme outer ones, like your skin cells and things like that, but pretty much all of the cells on the inside of you are floating in a fluid that is continuous with your blood. That is, your blood kind of passes into it and out of it, and it gets mixed constantly with your blood, okay? So that fluid is called extracellular fluid. It's the fluid outside of your cells. The idea here is that you need to pump certain things into the extracellular fluid, and you need to pump certain things out of the extracellular fluid into your cells in order to maintain homeostasis. By the way, we're almost done. This is the last page. There are two ways that your cells can do this, can pump things in or move things in and out of the cell. First of all, if they're going against the diffusion gradient, okay, that means that if you are, I'll give you an example. You've got a cell, and inside of the cell, you've got only a little bit of sugar, okay? So the sugar concentration is very low. These are my little sugars, okay? But outside the cell, the sugar concentration is high. Okay? When you have this situation going on, this is very easy for your cells because all they need to do is just kind of open a door and just let sugar come in through the door. Okay? Because this is with the concentration gradient. Sugar already wants to come in this scenario because of diffusion, right? So all you need to do is open the door, the sugar will come in. Oh, I just did this the exact opposite way. Oh boy, my apologies. I'm not trying to confuse you, I promise. So this right here is something that allows you to use passive transport. Passive transport needs no energy in order for it to work properly, okay? With the concentration gradient. because that's the natural way that it wants to move, right? This is like allowing water to flow down river. You're opening a gate and you're just letting the water flow down the river. So that's passive transport. So passive transport is when you're going down river. That's easy. You can either have a carrier protein. A carrier protein lets certain things just pass right through because of their shape. So in that sugar example, your cells have a sugar carrier protein that is the right shape to just let sugar go through them, okay? That's a sugar carrier protein. So if you have high blood sugar and you release insulin into your blood, which is what your pancreas does, then it just turns on the carrier proteins and they just let sugar flow into your cell, just going down river, okay? So that's one way that you can do it. Sugars always uses the example for carrier proteins, okay? It only works with particles that have no charge on them. If you have a charged particle, they can often be repelled by a carrier protein. They don't want to go near the carrier proteins. So this only works with uncharged particles. Sugar is uncharged, so it works really well with sugar. If you have a charged particle, like let's say that you want to let sodium or something come into the cell, you need one of these channel proteins. A channel protein is basically just a tube that allows charged particles to go through. It's an empty tube that has the opposite charge of the charged particle. So if you want to let in a positive sodium, okay, sodium is an example of a positive ion, then you just need a channel protein that is negatively charged. So it will basically pull, but passively, so it's not using any energy here, but it's allowing positive charges to just flow through it. It won't let negative charges go through though. Only the opposite charge to the charge of the channel. Yes?
What's the thing in the diagram?
Which one?
Like just the thing, like this too. Like where
The brown thing?
Yeah, where would that be? Like, what is
It's a protein and it's going across the lipid bilayer. So you can see the heads and the tails here for the lipids.
Where would that be in your body if you were just to like?
In the cell membrane. So this right here is the cell membrane. So these are running across the cell membranes for all of your cells.
Okay.
So for example, if I were to like zoom out here, if you had a cell, this protein, I'll do it in brown so to keep it consistent here, okay, would just be like in the membrane.
Oh, okay.
So it just allows charged ions to just go through. Okay, so those are called channel proteins. They allow charged particles to go through. And these ones that only let certain shapes go through are called carrier proteins. Both of these are passive, which means that they don't use any energy and they just allow stuff to go with the concentration gradient. Okay, so that's for the passive stuff. That's if you're going with diffusion. You're just letting stuff go through. But sometimes your cells don't want to just go with the flow. They don't want to just let stuff in. They actually have to pump stuff against the gradient. So a good example of that is when your cell makes waste products, there's always going to be very low waste inside the cell because you don't want no waste inside the cell. You want the concentration of waste to be zero. So in order to get the concentration of waste to be zero, you have to pump stuff. It's like a water pump. You have to pump the waste out of the cell to get the concentration as low as you possibly can. So in order to do that, against the diffusion gradient, you need an active transport pump. Okay? So in active transport, it works just like these. It's a channel that goes through it, but it's a pump. It physically grabs stuff inside the cell and pushes them outside the cell, and then grabs another one, pushes it out, grabs another one, pushes it out. So that's these active transport membrane proteins. So you've got passive transport proteins, those are carrier proteins, and channel proteins, which let stuff flow in the direction of the diffusion gradient. And then you've got active transport proteins that push against the flow. Those are pumps. So you can pump stuff wherever you want. If you need more of something outside the cell or more inside, you can pump it either direction with a pumping protein. Yes?
Can I fill up my water bottle?
Yes, you can.
Do I need a sign for that?
Actually, yes, that's also on the list. Sorry. Yeah. I think at pretty much any time you go, there's something on the list for that. Even if you're visiting your VP or something, that's on there too. Okay, any questions about this? I realize this is a lot of stuff we just covered here, okay? So, I'm gonna give you the rest of this block right now. It's until 12:50 to complete the first lab. I'm assuming people need time to work on the lab, right? Still? Okay, so I'm gonna give you that time to work on the lab. There is a lab activity that goes along with this. It's looking at diffusion in a little bit more detail. Okay, you're gonna do a little experiment with diffusion. How fast do things diffuse across a membrane? What speeds it up? What slows it down? That kind of stuff. We're gonna do that during Learning Block 1 tomorrow. Okay, so I'll have that lab set up for you tomorrow. It's actually in there now. I think it's scheduled for Learning Block 3. Let's see. Yeah, I have it there for Learning Block 3. So don't do the lab that's scheduled in Learning Block 3 today. We're gonna do it tomorrow morning. But if you have time during Learning Block 3, I would highly recommend that you watch that video of the person who drank too much water. It's a really cool video. So it talks about what this looks like in real life when you have the wrong concentration of solute in your blood. That is, you've consumed too much water. It's a pretty interesting medical case study. Okay, I'm gonna give you the rest of the time to finish the lab. Again, if you're in the chat here, I'll leave the chat open so you can leave your questions in there. You guys please ask your questions if you're getting stuck on anything, okay? Any questions right now, but you're okay to go? This lab needs to get handed in when you're done. Okay, so it gets attached to the assignment and then you just turn it in. Okay, I'll give you time to get started. And if you're looking for a copy of this note, it'll be in the course notes folder, which is also linked at the top.